Hello, my name is Massimliano Marvasi. I work at the Department of Biology at the University of Florence. And today I'm going to show a presentation about integrative application of microbial biotechnology. I've decided to show this type of presentation because I know the type of audience in this conference, which include engineers, designers and other type of professionals, and I think can be interesting to show how microbiology actually contributes to the development and the conservation of cultural heritage and the promotion. So basically today is a quick overview on three papers which give an idea how microbiology and similar disciplines are very important for the conservation of cultural heritage. Integrative from the Oxford Dictionary means serving or intending to unify separate things. And apparently microbiology may seem very different from a cultural heritage, a statue or portrait or a painting. Metagenomics also, for example, can be very far from a cultural heritage, but they aren't. Metagenomics, for example, is the whole genome, whole genes, whole genetic material on a cultural heritage, and microorganisms are everywhere, and so this DNA can be over any cultural heritage object, including a microbiota, which is the whole microorganisms, or the microbiome, which are also all the genetic information included in these microbes, and metabolomics also, which is the whole metabolites. And understanding the type of metabolites, for example, is very important because some metabolites can degrade the substrate, so they can damage the cultural heritage, or some metabolites, they can stain a substrate. So, as you can see, all these topics are related to cultural heritage, also biochemistry. For example, let's think about enzymes. There are many enzymes able to degrade cellulose, for example, and we know that many artwork are made of wood and cellulose. So all these topics are extremely important for the conservation and promotion of cultural heritage. In this slide, we can see the integration of these two aspects, so the cultural heritage and the microbiology component. Here we have a number of papers published in the field of cultural heritage conservation and life science in Tier 1 or 2 peer-reviewed journals, and the data has been obtained from Scopus and Web of Science. So you can see, as you can see, in the last 10 years, there has been a quite big increase of papers in this subject. This is very important because it shows the need of more integration and how microbiology can contribute to the growth of this field. Actually, this slide represents the keywords of cultural heritage and environmental sciences. In the next slide, I have here, in this slide, we have actually something more defined. We have the cultural heritage and metagenomics on the panel on the left, where we can see in the last years a big increase. Actually, this is not comprehensive at all. This has been taken in Scopus, but there are much more papers. So this doesn't really reflect. It just gives you an idea how this integration actually is increasing, but is not comprehensive. On the panel on the right, We can see the keywords cultural heritage and microbiota in Scopus, and we can see much more papers per here. Also in this case, this is not comprehensive, and actually there are much more, but gives an idea how this integration is more and more important. To show this integration, I will propose 3 different examples. There are 3 papers published by colleagues, and they show how it's important actually to study the microbial community present of cultural heritage. In this case, we have, you see these pits, these are from bricks belonged to the Auschwitz State Museum in southern Poland. And I got this interesting sentence from this paper, which say the characterization of microbial biodiversity and a sensitive degree of material biodeterioration requires a combination of methods. And in this very extremely true, because we are understanding how microorganisms with their metabolites and metabolisms are actually damaging the cultural heritage. So in this example, studying the brick specimens from this State Museum, from Auschwitz actually, so it's a very important museum in terms of preservation and valorization. This study is an example of conservation of degradation. Conservation and to study the degradation. In this case, the Association of Microbiology subjects use the study of the microbial community by using next generation sequencing, which is a methodology to understand the entire microbial community present on a substrate. In this case, we have the DNA extraction, as you can see in this figure, bacterial and fungal PCR amplification. It's a method to obtain specific genes related only to bacteria and fungi. And high-throughput sequencing is basically we can sequence everything, every gene related to the taxonomy of these microorganisms, and will result in obtaining the full view of the microbiota. Clearly, these give an idea of the type of microorganisms. When I know the type of microorganisms, I can understand if any of them are dangerous for the substrate. In this particular paper, which I found very interesting, there is an association to the microbiota, also the metabolome, so the entire metabolites. Also in this case, they have extracted the metabolites and they have performed metabolic profiles to see the type of degradation. These are cyanobacteria. In this slide you can see a layer of cyanobacteria. You can see how they are over the surface. You can see how they degrade like this. You see the tip. This is basically has been generated by the cyanobacteria. Clearly, it would be very interesting to know the type of metabolites involved in this degradation. Yeah, in this slide we can see some of the metabolic profiling that show the type of pathways that can be found on this cyanobacterial layer. Clearly, these pathways are not only limited to the cyanobacteria, but is the entire metabolome. The researchers study the entire metabolome, and by their extension, they use bioinformatics software to figure out the entire pathway, metabolic pathway. As you can see here, there are is interesting because there are several types of pathways. I would like to show your attention, to bring your attention to the point 8, for example, on carotenoid biosynthesis. Carotenoids are also able to stain, for example, the substrate. So it's not only a problem of degradation, but also about staining, as we mentioned before. We will see also later something similar in another example. So the Understanding the metabolic pathways on cultural heritage is important for understanding acid metabolites, production of pigments, degradation pathway of, for example, chemical treatments. For example, if I put a glue on a cultural heritage, this glue can be degraded by other microorganisms, so the treatment is basically wasted. Or degradation pathways of the actual artwork substrate, as we said before, for example, in case of cellulosic bacteria able to degrade cellulose, and so this is why it's so important to understand this type of metabolites. In this example, this is a second example where the authors, in this case, they have studied the biodeterioration process of building materials. Biodeterioration is not only about degradation, but can be also staining. So some pigments that are able to change the color of the artwork. In this case, we can see they have done an interesting experiment on salt crystallization. You know that in bricks, for example, it's easy to form a crystallization due to the capillary suction, for example. And the bacteria can actually grow on these crystals structure and stain, because they can produce pigments. The metabolism of halophile are microorganisms able to survive and replicate in a high concentration of salt. So the halophile metabolome was therefore studied in this paper by using different salt solution. So basically they simulate different salt degradation on bricks in order to see which type of bacteria can grow and replicate. This is in this slide is salina, which is an environment used to produce salt from the sea water. And this is from Cabo Rojo in Puerto Rico. You know, Puerto Rico is a very hot climate, hot weather, so the evaporation is very fast, and it's possible to recollect salt. If you see the salt, is this square part on the picture are different area where the salt evaporates at different level of evaporation. And the interesting thing is the color. They are pink or red, and this color is due to these halophiles. So microorganisms able to survive in high concentration of salt, they actually produce a lot of pigments, so high producer of pigmentation. That means also staining on the on the bricks or the cultural heritage where they can grow. In this experiment, the researchers have done a mock experiment where they study the formation of crystals on bricks of different type of chemistry. As you can see in the table on the left, there are different type of salt, and they have then grown different microorganisms isolated from previous stone material from cultural heritage, and then in order to see, to test, to check actually different metabolomes, different type of metabolites. And you can see in panel A, B, and C actually are the different type of metabolites based on a PCA analysis, and how actually even type of metabolites clusterized together according with the type of salt used to grow the microorganisms. In some of these examples, beta-carotene was also present. Again, we saw also in the paper before, and showing how actually this microorganism can stain the substrate. But this is interesting, showing that also with the metabolome we can clusterize, we can understand the type of bacteria also growing on the substrate, and then we can understand and see if any of these metabolites may be dangerous for the substrate. This is the third example, where we can see how microorganisms can not only degrade a substrate, but they can also restore a substrate. This is a fresco from the Camposanto Monumentale in Pisa. And in this case, if you can see in the panel above, is quite degraded, and this degradation is due to a patina of a glue used to remove the fresco using a technique, say the strappo, which is a technique to remove the fresco from a wall, and was used during the Second World War to protect this type of fresco, to remove them quickly from the wall. And the problem was that the glue, actually, after decades, was fixed on the paint, and as you can see on the panel above, was covering the painting. Basically, in this study, realized in 2004, so it's quite old but it's still a very actual paper, you can see how degradation, how these microorganisms can degrade the glue, and the result are in the bottom of the panel, so in the bottom panel, where you can see the figure is completely visible. Now it's clear, we can see it very easy. And in this case are microorganisms. This table is the type of microorganisms used, and I would like to bring your attention to the Pseudomonas tutzeri, A29 strain, which actually was the strain used to achieve this greater result, is a strain able to degrade protease. So with a high protease activity, because I forgot to mention that this glue is basically formed by animal glue with a high content of proteins. So also in this case, you can see how microorganisms, this type of microorganisms are able to degrade the glue and release a complete clear picture by removing the patina. So basically there are many future challenges in the association of microbiology and cultural heritage. It is still a lot of work is to do, is still needed, but I think that microbiology and related science can really boost the conservation of cultural heritage. So if we understand better the microbial ecology on substrate, we can improve the understanding of biodeterioration processes, and possibly to stop them, we can apply better the restoration treatments, for example, by the analysis of metabolites. We can better understand the microbial community and fluctuation of them, considering that there are some microorganisms, they can stain the substrate because they produce pigments. So we can, for example, by understanding the fluctuation, so how microbial community change over the time, we can possibly stop this type of microorganism that degrade the substrate. And there are also new technologies like the EVA technology or EVA technology, which is lately presented by Professor Righetti, which is a great technology to study, to promote cultural heritage. And finally, biorestoration. I showed you an example about biorestoration, but there are also other examples. For example, the use of bacteria able to precipitate calcium carbonate for stone consolidation. So with this brief overview, I hope I showed you some example on how microbiology can be used in cultural heritage with a little bit focus on the latest technique of on metabolomics and biorestoration. And I would like to thank you very much for your attention. Thank you.